Hey everyone, today we're diving into the nervous system, the body's command center. Think of it like a superhighway. We have two main divisions: the central nervous system, CNS, which includes the brain and spinal cord, the main control center. Then there's the peripheral nervous system, PNS, which are all the nerves that branch out to the rest of your body, like the roads that lead to different cities. The nervous system is responsible for coordinating bodily functions, processing sensory information, and enabling cognition. Neurons are the key players in the nervous system, acting like tiny messengers. They use electrical signals to communicate with each other. A neuron has three main parts. First, dendrites, which receive signals from other neurons. Imagine dendrites as antennas picking up messages from nearby cells. Second, the cell body, which keeps the neuron alive and processes the signals it receives. Third, the axon, which sends signals to other neurons. Imagine axons as transmitters sending messages to the next cell. Watch the action potential travel along the axon. Key takeaways. Neurons are messengers using electrical signals. They have dendrites to receive, a cell body to process, and an axon to send signals. Action potentials travel along axons like messages being transmitted. Neurons get a lot of support from glial cells, which are like the backstage crew keeping everything running smoothly. First up are astrocytes. These star-shaped cells provide nutrients to neurons and help maintain the blood-brain barrier, which protects the brain from harmful substances in the blood. Next, oligodendrocytes and Schwann cells are the insulation experts. Oligodendrocytes wrap myelin around axons in the central nervous system, like the brain and spinal cord, while Schwann cells do the same job in the peripheral nervous system, covering the nerves outside the brain. This myelin sheath acts like electrical tape, speeding up signals and preventing energy loss. Microglia are the brain's immune cells, always on patrol. They clean up dead cells, fight off infections, and remove harmful substances. Think of them as the security guards that keep the brain safe from threats. Finally, ependymal cells line the ventricles inside the brain. They have tiny hair-like structures that help circulate cerebrospinal fluid, which cushions the brain and removes waste. It's like having a built-in cleaning system. So remember, while neurons get all the attention for signaling, neuroglia are the unsung heroes that make it all possible. Before a neuron can send a signal, it has to be ready. This ready state is called the resting membrane potential. It's like a battery waiting to be used. The inside of the neuron is more negative than the outside, usually around negative 70 millivolts. This difference is maintained by the sodium-potassium pump. It uses energy to pump sodium ions out and potassium ions in. This pump's energy expenditure accounts for about 20% of the brain's ATP use. Now for the fun part, the action potential. This is how a neuron sends a signal. When a neuron receives enough stimulation, it depolarizes. Voltage-gated sodium channels open, allowing sodium ions to rush in, making the inside more positive. Then, sodium channels close and potassium channels open, letting potassium ions out, bringing the neuron back to its resting state. This rapid change in voltage creates an electrical signal that travels down the axon. Refractory periods prevent refiring, ensuring unidirectional signal propagation at speeds up to 120 meters per second in myelinated axons. Key takeaways. Depolarization brings sodium ions in, repolarization sends potassium out, the signal travels along the axon, and refractory periods keep it going in one direction. The action potential travels along the axon and reaches the synapse, the junction between two neurons. Here, the electrical signal needs to jump across to continue the message. Most synapses are chemical synapses. When the action potential arrives at the synapse, it opens voltage-gated calcium channels. Calcium flows in and triggers vesicles to release neurotransmitters into the synaptic cleft. The neurotransmitters cross the synaptic cleft and bind to receptors on the postsynaptic neuron. This can excite the neuron, increasing the chance of an action potential, or inhibit it, reducing that chance. While chemical synapses are the most common, electrical synapses connect neurons directly through gap junctions. Ions flow straight through, allowing rapid, synchronous signaling. These are found in places like the heart or retina, where speed matters. Key takeaways. Synapses bridge neurons, chemical synapses dominate with neurotransmitters, and electrical synapses offer fast, direct signaling. Sometimes things go wrong with the ion channels. These malfunctions are called channelopathies. For example, in cystic fibrosis, there's a defect in chloride channels, causing mucus buildup in the lungs. In some types of epilepsy, there are problems with sodium channels, leading to seizures. Congenital insensitivity to pain can happen because of sodium channel mutations. Channelopathies affect about 1 in 500 births, according to NIH data from 2025. Let's look at some real-world examples of channelopathies and their treatments. Triple CFTR modulators have restored lung function in 85% of cystic fibrosis patients by fixing chloride channel function. Gene therapy targeting SCN1A mutations in epilepsy has normalized seizure thresholds in mouse models and reduced status epilepticus episodes by 60% in human trials. A 25-year-old with an SCN9A mutation causing congenital insensitivity to pain experienced repeated fractures from undetected injuries, leading to amputation. Looking to the future, we see exciting trends like personalized medicine, neuroengineering, CRISPR gene editing, AI integration, and brain-machine interfaces. It's a very exciting and promising future for treatment of the nervous system.